I am your facilitator, Dr. Danielle N. Gray Singh. I have been a member of the Capella family for 2 rewarding quarters and am eager to begin my 3rd quarter with you. My husband and I agree that Capella affords me the opportunity to try to be a supermom to our 2 little slices of heaven. They are quite the little scientists. By way of formal introduction, I earned my Bachelor's of Science degree in mathematics with a concentration in computer science and chemistry from Xavier University. I have an earned doctorate in neuroscience from Tulane University, and finally I pursued my postdoctoral studies at Harvard University's MGH East's Genetics and Aging Unit and at Baylor College of Medicine's Huffington Center on Aging. Since 1997, I have been exploring the neurochemical events that are responsible for the onset and progression of Alzheimer's disease. My research efforts have produced an interesting picture about the role of metals in the formation of the hallmark feature of AD, plaques. Plaques are sticky masses consisting of amyloid protein. The normally soluble amyloid proteins bind to biometals or metals and fall out of solution, forming insoluble plaques between neurons. It was once thought that the commingling of amyloid and metal was irreversible. However, my research efforts demonstrated that by extracting or pulling the metals from the plaques could resolubilize the amyloid. By using metal chelators, I was able to do just that in mice. My results were considered groundbreaking and led to 2 clinical trials involving humans. To date, the drug studies have been an abysmal failure. C'est la vie. Nonetheless, my research efforts have earned me a spot in the intellectual lineage of Dr. Colin Masters, who discovered the APP gene, and my direct academic advisor at Harvard University, Prince Ashley Bush, who discovered in 1992 that zinc could induce plaque formation. Dr. Bush challenged me to discover if these same events took place in mammals, so I investigated the role of metals in AD in humanized mice overexpressing the human APP gene. My most recent work attempts to shed light on the role of neurosteroids in AD. It appears that the role of sexoids in the aged brain is to recall the neuronal stem cells from their slumber or senescence and move them back through the cell cycle. This is very intriguing because it suggests that humans may actually have the power to regenerate previously lost cells or neurons. Now, the million-dollar question is, how can we unlock that potential? It would be awesome if I could have you engage me in this mental exercise to find a way to do just that.